Okay, so I'm Ivanka. I'm a high school student who's really passionate about climate and sustainability. And I first got interested in methane emissions when I did my research project with the University of Minnesota and also at Cornell University in reducing methane emissions from livestock. And so I started this initiative to educate people on methane emissions, like its sources and then impacts and some mitigation strategies. And then I also wrote this book on methane emissions.
Yeah, I saw that from your other podcast. That's amazing.
Yeah, thank you. Yeah, and that goes a lot more into detail about methane emissions. Yeah, I'm really excited to have you on the podcast today.
Well, thank you for having me at your podcast.
Yeah.
So you want me to introduce myself?
Yeah, that would be great.
Okay, so I'm Ran Mei. I'm an assistant professor in civil and environmental engineering at the University of Illinois at Urbana-Champaign. So my research is about recovering methane from wastewater streams and use methane as a renewable energy source. Yeah, so it's slightly different from other guests at your podcast because they want to control methane emission,
Yeah.
But at my space, we want to stimulate increased methane production from wastewater.
Yeah, that's really interesting. I'm so excited to learn all about it. Yeah, okay, so can you share a little bit about your educational background and how you got started?
Okay, yeah, sure. So I had a bachelor's degree in energy and resources engineering from Peking University in China. I had my master's and PhD from this department at the U of I, and my graduate work was about methane production from wastewater. After that, I went to Japan. I spent 3-4 years there studying the evolutionary history of methane-producing microbes. So how did those microbes acquire this very unique pathway and capacity, and how was this pathway inherited between different microbes? And after that, I came back last November, and so I have been in this position for about 10 months. So I'm still in my rookie year in my assistant professor role.
Okay. Yeah, can you talk about some of the projects you're working on?
Sure, yeah. So here I'm based in this environmental engineer department. So I work on quite broad aspects of water engineering, but the project that is most related to methane is to study how microbes that produce methane coexist with other microorganisms, because in natural environment and also in engineered environment, like a water treatment plant, wastewater treatment plants, microbes always live with other species. And for those who can produce methane, they have to rely on other bacteria to pass on important food to methane producers. And this collaboration is very important for many wastewater treatment processes, and in many cases, it's also very sensitive to the environment, like to temperature and to pH. So the project I'm very excited about is to figure out the optimum environment for this species interaction. So how can we protect this relationship so that both partners can be very happy, so we can get as much methane as possible.
That's interesting. So what's like the next step of that research then?
Okay, so my research group here at the U of I is very young and we are still developing. So for this project, we are at the very beginning of a big picture. So in wastewater treatment plant and also in human and in natural environment, the microbial community is very complex. Usually like hundreds or thousands of different species coexist. So this complexity brings a lot of difficulty in studying them because they are living together and they're very tiny. So it's almost impossible to separate them. But a step back strategy is to use some model organism and study them in lab. For example, my current experiment is focusing on just a small bottle that only has 2 species instead of thousands of species. So with this 2 species ecosystem, we can understand some of the most fundamental and important pathways that will have an influence on methane production. And after that, we can maybe study a 3 species environment and maybe a 4 species environment, and eventually to a realistic environment where we can see how can we produce more methane, how can we treat wastewater at a higher efficiency. So I'm currently at this step 1, a 2 species glass bottle that is relatively easy to study.
Okay, that's cool. So what 2 species are you looking at?
Okay, so one species is a methane producing organism, so we call them methanogens. And we are currently using a very model methanogen because the genome of this methanogens has been sequenced, so we know quite a lot about its genetic capacity and its physiology. So the other partner is a bacterium that can produce hydrogen and formate. And hydrogen and formate are important because they are the food to methanogens. So methanogens will convert hydrogen and formate to methane. So how do they exchange this intermediates? So the hydrogen and formate is a critical step in this entire methane production process. So more importantly, they are different partner bacteria. Some can produce hydrogen at a higher rate and some producing hydrogen at a lower rate. So those complexities also part of our research. We want to figure out between different partners what their, for example, favorite temperature and what's their favorite pH and favorite other physiochemical parameters.
Okay, that's really interesting.
Yeah.
Are you doing any other projects?
Sure, yeah. For example, I'm also very interested in the evolution of microbes. So when I was a postdoc, my research project was to study, for example, when did the first microbe that can produce methane emerged. So, and after analyzing genes and comparing the genomes of methane producers and non-methane producers, our conclusion is that the first microbe that can produce methane appeared on this planet about 3.7 billion years ago. So 3.7 billion years ago. So that's a very long time ago and almost just a little bit later than the first cell appeared on Earth. So methane production is a very ancient pathway, and it played a very important role in the formation of more complex cells. For example, like human beings, we are multicellular organisms, and our ancestor, based on the theory, our ancestor was a fusion between one microorganism and another microorganisms. And the microorganism that provides the cell envelope is kind of related to methane producers. So methane production is not only an important pathway, it also it was a very important process during the evolution of all other kinds of life. So now in my new position here, I'm interested in how, for example, bacteria learned to degrade micropollutants, especially pollutants that is synthesized by human beings. For example, pesticide, antibiotics, and pharmaceuticals, personal care products. So those chemicals are mostly synthesized by modern chemistry, and microbes have never seen those chemicals in nature, right? But how can we train microbes to degrade those chemicals so that those chemicals won't become a toxic pollutant in the environment? So I'm also looking at evolution at a modest scale, like within several decades, can we invent new microbes for waste cleanup?
Yeah, what kind of chemicals are you like looking at?
Yeah, so for example, in our preliminary analysis, we are focusing on a chemical called terephthalate. So terephthalate is a monomer to synthesize plastic bottles. So almost every plastic bottles that we use today is synthesized using the polymer of this aromatic compound called terephthalate. And terephthalate is mostly synthesized by modern chemical industry. However, and this chemical was patented in the 1970s, and so it's not that long, just maybe half a century ago. However, we already know two bacteria that can degrade this very reconcentrated, very strong chemical. So when did they learn to degrade this new food, new substrate? Based on our genetic analysis, it turns out that about 30 years ago, which is roughly when I was born, this bacteria learned this new pathway in wastewater treatment bioreactors.
Oh, okay.
And the reason they can learn this new pathway is because wastewater treatment bioreactor has a very high concentration of pollutants compared to a river, to a lake, or to the ocean. So the high concentration of those contaminants is a very strong selective pressure for bacteria. So if you're a very smart bacteria, if you learn things very fast, you can survive in this very unique environment, and you will become the dominant species of this ecosystem. And my hypothesis is that in wastewater treatment environment, there are likely many new evolutions where bacteria learned genes and pathways to degrade new chemicals that should be cleaned up before the wastewater is discharged to the natural environment. So I'm currently working with my graduate student to look for those new genes, new proteins, and for longer run, we hope we can apply these new proteins, new genes to for bioremediation, for example, to clean up waste in soil, in water. Especially a lot of the chemicals are very hard to be cleaned up using chemical or physical methods. So that's another project that I'm working on right now.
Okay. Wow. Are there any other projects you want to share?
So I also collaborate with other faculties on this campus and also in other universities. For example, a faculty here in our department is applying AI. So I saw this question in your question list, how do we apply AI in environmental engineering? So she's applying AI to identify new genes that can depolymerize, basically break down plastics. So most plastics are polymers, so like hundreds and thousands of chemicals linked together. But the first step to recycle plastics is to break down the polymers into monomers. And AI is very powerful in identifying new genes and enzymes that can be used for this purpose. So that's one project I'm collaborating with other faculty. And I'm also working on degrade an algal species that can degrade plastics, because plastics is everywhere, and it's a really important environmental issue. And not only bacteria, but other organisms can also grow and degrade plastics. So I'm applying some genetic analysis to this organism, and hopefully we can apply this organism into wastewater treatment, into bioremediation, and in other scenarios.
Okay.
So basically, I'm applying genetics and microbiology to different environmental engineering environment. Water, primarily is water, but also soil and like agricultural waste.
Yeah, that's really cool. Okay, so what's your, who all is your lab and team made up of?
Okay. My lab is still in our first year, so we have only one PhD student and one master student, so just 3 of us. But I hope we can expand very soon in the future, especially if it will be great if we can have like undergraduate student or even high school student participating research to address important environmental issues. But currently we are a very small, very young group.
That's cool. Yeah, if I went to Illinois, I would love to join your lab.
Oh, that's great. And I actually watched some of your podcasts, and you're very motivated to do like climate change research and
Mhm.
also sustainability. Here in our department, we have a program called EWS, so Environmental Sustainability and Water Resource. So it's an interdisciplinary program that covers some of basics in environmental engineering, but also most advanced topics in sustainability. So if you are interested, definitely you should consider us.
Yeah, that sounds great. Okay, so are all your projects like mostly in the US or do you do projects in other countries?
Yeah. So I have a collaborative project with my colleagues in Japan. So
Okay.
that project, oh yeah, I forgot to mention that. So that's also related to methane because I'm working primarily on methane producing microbes in engineered environments, like in wastewater. However, those microbes are also very common in other environments, for example, in freshwater sediment and also in marine. So I'm doing a genetic analysis, comparative genetic analysis to see whether methane producers are using different strategies in those different environments. Do they have do they share the same lifestyle, or they have different pathways and ecology in marine, in freshwater, and also in bioreactors. So, and that is in close collaboration with my colleagues in Japan.
Hmm. So is it at a university in Japan or?
No, it's at a research institute in Japan.
Okay.
So the Japan Agency for Marine Earth Science and Technology.
Okay.
Yeah.
By any chance, are you also doing anything in like rice cultivation method?
No, I'm not doing anything about in that aspect. But here
Okay.
on this campus at U of I, we have a very strong department of animal science and also agricultural science, and there are outstanding researchers doing methane emissions from agricultural waste and also from cattle, from rumen. Yeah.
Yeah, that's good to know. So are there any other universities you're collaborating with in the US?
So the algae that can degrade plastic project is a collaboration with a professor at the Southern University at Southern Illinois University at Carbondale.
Okay.
And and I'm also, it's not a formal collaboration, but I'm working with a professor at Montana State University to study a very new microbe that produces methane. So he identified this very unique microbe in, I believe it was in Yellowstone, the nature park, the national park, Yellowstone National Park. And by analyzing the genes and genomes, he found that microbe is also present in wastewater treatment environment. So we are at a very early stage to look for if we can find more methanogens, methane-producing microbes in wastewater treatment environment.
Okay. That's interesting. Are you collaborating with any like private companies or maybe like a government department in the US?
Currently, no, but when I was a graduate student here, my almost the entire period of my graduate study was supported by two companies in the industry. So they are, so one of them is a petrochemical company that produce a lot of wastewater, and they use a bioreactor to treat their wastewater, and they also recover methane from that bioreactor to provide renewable energy for their plant. And the other company is producing, it's a leading manufacturer of a soft drink. And they also generate a massive amount of wastewater with very high concentrations of carbon and nitrogen and other nutrients. So I was also working on a bioreactor to clean up their wastewater and also recover methane as a renewable energy.
Okay.
Yeah. And in the future, I definitely want to apply my findings in microbiology, in engineering to the industry.
Yeah. So what are some challenges you face in these projects you're doing?
Yeah, So before I started this position, like many of my mentors and colleagues already told me that it's gonna be difficult at so many times. But since I started last year, I feel the challenges are beyond my expectation. For example, nowadays it's so for example, ordering chemicals is increasingly challenging because some of chemicals are imported from other countries and we are facing like tariffs and stuff.
Okay.
So that's a new challenge. But overall, it's going well. This is a great department. I like people here, and faculty and staff are very supportive. So they help me to grow as a junior faculty. So there are challenges, but I'm optimistic that those can be overcome. Yes.
Yeah. So in your work, do you spend more time in the lab or do you go to the field, or like where do you spend your time?
So my research is a blend of wet lab experiment in the lab and also data analysis in computers. So in the lab, we grow microbes, including those that can produce methane in like small glass bottles. I also operate lab scale bioreactors, so like 5 liter, 7 liter, 10 liter bioreactors in my lab to mimic a full scale wastewater treatment environment.
Okay.
Because the lab scale bioreactor is more controllable, we can do whatever we want to the reactor. But the full scale reactor, they have to meet permit and they have to make sure that it's operated correctly. So a lab scale bioreactor gives us a lot of flexibility to change the operating condition and so that we can test our hypothesis. I don't do a lot of sampling. The only place I go is wastewater treatment plants. For example, in Central Illinois, I went to like over 10 wastewater treatment plants and collect samples from their processes. And I bring a sample back to the lab and analyze their primarily analyzing their genomes. That's where I do analysis on computer.
Okay. So you get all your microbes from the wastewater treatment plants directly?
Yeah, pretty much. Yeah.
Okay. Wow. That's cool. Okay. And then what advice would you give someone like me or a high schooler who wants to go into a field like this?
Okay. I think first of all, keep curious. I think you did a very good job to show your motivation and curiosity, like why methane is important and how can we either control methane or promote methane in other scenarios. So if you lose your curiosity, I guess the chance of achieving something important will be very low. So I think especially if you want to stay in research, in science, in academia, being curious is very important. And in addition to that, I think for a high school student, maybe build a very solid foundation on like math, physics, chemistry, biology, and maybe a little bit coding because AI is so important today. Because in the future, most research you're gonna do will be interdisciplinary. Some chemistry, some biology, some math. If you can keep your fundamental knowledge with you throughout your career, that will be super helpful. Yeah, so
Okay.
don't make a choice too early. Don't say, okay, I will only do biology, so I will just skip all the math and physics. I think maybe that's something you don't want to do at high school.
Yeah, that's good advice. And also for growing your lab, what kind of skills would you be looking for?
So my current project involves a technique called anaerobic cultivation, basically how to grow microbes, for example, methane producing microbes in lab in a small bottle. It sounds very straightforward, but actually when you practice it, it's quite difficult because for microbes that can produce methane, they require a very strict anaerobic environment, so no oxygen. Like for human being, we need oxygen all the time, but for those microbes, oxygen is toxic. So how to provide a small environment where there's no oxygen is a little bit tricky. So for example, my postdoc advisor in Japan, he spent like 20 years growing one particular organism. And of course, the result was groundbreaking. It was published on Nature, and it's got like thousands of citations. But just to grow that particular microorganisms is a very tricky practice. And we also do some basic molecular biology experiments like DNA extraction, like PCR. We also do some microscopic examination. Meanwhile, we do bioreactor operations, so small reactors in the lab. And that also takes a very long time because you want to check your reactor every day and make sure that there's no leaking, temperature is all right, pH is okay. So it's a lot of work. So I have to acknowledge my student who's doing that work now.
Okay.